So welcome to this webinar organized by EURO-NMD with the Rare Neurological Disorders ERN and the European Academy of Neurology. It's CMT month in October 2023, and today we have the pleasure of having with us Professor Kleopas. Kleopa is the head of the Neuroscience Department, coordinator of the Neuroscience Master and PhD graduate program of the Cyprus Institute of Neurology and Genetics, and he also coordinates the clinical center dedicated for neuromuscular disorders. And besides his interests in the neurological and neuroscientific fields, he has an acute knowledge of targeted advanced therapies, namely gene therapies for inherited neuropathies and local dystrophies. He is a board member of the International Charcot-Marie-Tooth and Related Disorders Consortium and serves as a member of the Scientific Advisory Board of the USA CMT Association and of the Gilbert Foundation Neurofibromatosis 1 Gene Therapy Initiative. So it's a great pleasure to have him with us, and I pass on the word to David Peirison, our chair of the working group on peripheral nerve, to give us some information about today's webinar. David?
Thank you, Antonio. Thank you very much, Kleopas, for being here today. This month, October, is the month of CMT awareness. So it's important that we are having 4 webinars about CMT, 2 in the last weeks, one today and one next week, given by Sharamatarian on diagnosis of CMT. Kleopas is a important member of the management group of the ERN Neuropathy Working Group. And also it's a real pleasure to have him today as a speaker because he has pioneered important papers and work on gene therapy for CMT. Thank you, Kleopas.
Thank you very much, Antonio and David, for this kind introduction. It is my honor and pleasure to give this presentation today for the ERN EURO-NMD as part of the CMT Neuropathies Month. I will talk about the gene therapies. Some of them developed by my group, but I will also try to give a more overall view of the field and also touch upon various developments by other groups for various types of CMT neuropathies. You don't probably need much of the introduction, but I would just like to show this nice figure from David's paper showing the phenotype of CMT neuropathies. And as you all know, is the CMT neuropathies are the most common neurogenetic disease with a collective prevalence of 1 in 2,500 individuals worldwide. The onset is typically in childhood, latest in adolescence, but there is a wide range of disease onset from infancy to old age. It's a very variable phenotype and age of onset. And the presentation is typical with slowly progressive weakness and atrophy of the hand and leg muscles, loss of sensation, impairment of balance, impairment of walking. Unfortunately for us trying to develop treatments for this disease, the disease is highly genetically heterogeneous. There are more than 100 different causative genes, and you see here in this nice diagram from Vincent Timmerman's paper, they're spread all over the genome. And so that creates a challenge both in the diagnosis, but also in the development of treatment strategies. And so most of these neuropathies will require an individualized and disease-specific approach. In order to discuss therapy developments for CMT neuropathies, it is important to first have a basic understanding of the cellular and molecular mechanisms that lead to the disease. And the way that we understand CMT neuropathies is by dividing them into axonal and demyelinating forms. Axonal CMT neuropathies, also known as CMT type 2, are caused by mutations and dysfunction of genes that are expressed in neurons. And these genes play important functions in neurons and the axons that innervate the muscles or carry sensory signals to the body. And you can see here a variety of responsible genes, including several that regulate mRNA processing and protein translation, several genes that are essential for axonal transport, including mitochondrial proteins, mitofusin 2, GTAP1, ion channels, even the neurofilaments themselves that are part of the axonal cytoskeleton. We then have the demyelinating CMT neuropathies, which are the most common, and they typically result from dysfunction of genes that are expressed by myelinating Schwann cells, and these are classified as CMT type 1 or CMT type 4, type 1 being autosomal dominant and type 4 being autosomal recessive. And you can recognize here in this diagram of the Schwann cell and the myelin sheath some of the most common CMT genes, including the peripheral myelin protein 22 and myelin protein 0, both components of the compact myelin, as well as connexin-32, which is a component of the non-compact myelin. And along with this, we have several other genes related to cytoskeleton, to the extracellular matrix, but also to endosomal sorting, signaling, and mitochondria function as well. Another important aspect that I would like to emphasize before I move on to the treatments is the challenges we face with this disease. And two of the major challenges is the rarity of most types and the slow progression. And that's important when we try to develop treatments. So among the more than 100 different CMT genes and subtypes, most are very rare, with the exception of CMT1A, which is the most common type and makes more than half of all cases, estimated the prevalence of 1 in 5,000 individuals. Second most common is the CMT X-linked type 1, which is estimated to have a prevalence of 1 in 25,000 of the population. And from there onwards, we have very rare CMT neuropathies. The next most common, CMT2A and CMT1B, 1 in 36,000. The SORT neuropathy, which has emerged in recent years as a relatively common one, has a prevalence of 1 in 100,000. CMT4C is less than 2% of all CMT types, and CMT4B less than 1%. So, and the other ones are even more rare. So this results in difficulty recruiting patients, for example, for natural history studies, and also for potential treatment trials. And also the slow progression creates challenging in developing treatment trial designs, in having adequate measures to evaluate disease progression, and that creates also the need for biomarkers. I will not get into detail about this, but this is part of the challenge that we face when we develop therapies. So in terms of the treatment for CMT neuropathies, as you all know, we have a long pathway towards approved treatments that starts from understanding the disease mechanism. So that involves the knowledge of the gene function, the knowledge of the cellular and molecular effects that the mutations have, most often tested initially in vitro. Then we have to move into in vivo models because they have myelinated nerves, and we have to test treatments on myelinated nerves. And of course, before moving to any type of translation, we have to scale up treatments in larger animals, most typically non-human primates. And only when demonstrating efficiency and safety in larger animals, we can move to clinical trials. And they can take also a long time, moving from phase 1 to phase 4 before hopefully we reach the stage of approved therapeutics. Where do we stand today with CMT neuropathies? The drug therapy space is more developed. So currently we have at least 2 drug treatments that are in clinical trials. One is the PXT drug therapy for CMT1A, and the other one is the aldolase inhibitors for SOD neuropathy. So these have already reached phase 3 clinical trials. And when we talk about gene therapy, we're even much more behind. So we have reached the stage mostly of rodent model testing. Some of these gene therapies have reached non-human primates, and almost none of them has reached clinical trial, with the exception of CMT2S, which is a gene replacement therapy for a very severe infantile form, actually the SMA phenotype of this gene. And I will say a few words about this. So overall, we have not made much progress in terms of having approved therapies. There is currently no EMA or FDA-approved gene or drug therapy for CMT neuropathies, and all that we offer to our patients, as you know, is supportive treatment at this point. Hopefully, this will change in the near future. So I will focus now on gene therapies. And so when we talk about gene therapies, we have to, first of all, think about the disease mechanism. And I divide here the disease mechanism basically into loss of function and gain of function. So in the loss of function examples, we have loss of the gene function, and in this case, we have to replace the normal gene in the cell that is requiring its function. In the cases where we have a gain of function, and that's actually many of the most common CMT neuropathies, we have to develop other strategies. We use gene silencing to reduce toxic levels of the gene, or we have to use allele-specific silencing or gene editing. And I will show some examples of these approaches further down in my talk. The other important consideration is that we have to deliver the gene therapies to the affected cell type, as I described before. So in the case of demyelinating CMT neuropathies, we really have to deliver the therapeutics to myelinating Schwann cells in order to restore function or to stop the toxic effects in Schwann cells. And the challenge with this is that Schwann cells are spread throughout the peripheral nervous system. I will come back to this issue. In the treatment of axonal CMT neuropathies, we need to deliver the therapeutics to the neurons because these are the cells that express the relevant genes, and this is how we can get our treatments down the axons and hopefully restore the disease mechanism. So that's very important because that determines also the delivery approaches and the delivery vehicles used for gene therapies. A particular challenge has been the targeting of Schwann cells. And this is because Schwann cells are distributed throughout the length of all our peripheral nerves that are found all over the body. So in contrast to targeting neurons to treat axonal neuropathies, this is a much bigger challenge in terms of the biodistribution and effective cell targeting. The neuronal targeting has already been partly solved, and this has been very much pioneered by the example of the SMA gene therapy, because we have an efficient way to target the motor neurons, for example, with viral vectors, at least in infants. It remains a challenge, of course, for older patients, but for Schwann cells, it has been a real challenge because we really need to get our gene therapies as widespread as possible in the organism with a safe and clinically applicable approach. So various delivery methods have been tried, various injection methods, and I show here a few. There has been, for example, an approach directly injecting into the nerves, mostly the sciatic nerves. We have tested this, and other groups have tested this approach, and that results in gene expression in the injected nerve. It's quite localized and doesn't really serve to solve the problem in a more widespread approach. The intramuscular injection it has been used mainly to deliver trophic factors, which are supposed to have an indirect effect on nerve integrity and support nerve function, but do not really get any therapeutics to the nerves themselves. The intravenous delivery is an approach that may be clinically easy to use, but will require bigger amounts of therapeutics to reach the nerves and potentially have more toxic effects. And so, at least in my group and in our programs, we have focused more on the lumbar intrathecal delivery route, so delivering directly into the CSF space. And we found that this approach can provide the more widespread targeting of the peripheral nervous system with relatively lower doses of the viral vectors compared to, for example, intravenous delivery. Another important, and of course, the vehicles used for gene therapy are typically viral vectors. Currently, the leading viral vector for in vivo gene therapy are the AAV vectors, or adeno-associated viral vectors. We also used in the past lentiviral vectors, but because of integration into the host genome, they are less attractive for in vivo gene therapy. And they're mostly used now for ex vivo gene therapy, which is not something really relevant for CMT neuropathies. And there is now also an effort to develop non-viral vehicles to deliver therapeutics. These are mostly relevant for RNA therapeutics, and I will show some examples, but there is also an effort to use them for delivering therapeutic DNAs as well. But this is only at the beginning, but it includes nanoparticles and extracellular vesicles and other approaches that are currently in development. In all of these cases, whether it's a viral vector or a non-viral vehicle, it is important that this vehicle is able to enter into Schwann cells. And that means that they have to have a tropism, they have to get through the blood-nerve barrier or through the basal membrane and through the Schwann cell membrane to introduce the gene, the therapeutic nucleotide into the Schwann cell. So both the biodistribution and the Schwann cell targeting are two important requirements for having effective gene therapies, at least for the demyelinating CMT neuropathies. So with all this introductory background, I will start with the most common CMT type, CMT1A, which is very well known to everybody. This is a disease that results from a duplication of the peripheral myelin protein 22 kilodalton gene, and this is because of an unequal crossing over during meiosis. So it results in 3 copies of the gene in individuals affected with CMT1A, and this causes an overproduction of PMP22 protein. It is clearly a gene dosage effect. The PMP22 is an integral protein of the compact myelin, but seems to have also important regulatory function in Schwann cell growth and differentiation. And it is necessary both for the formation of myelin during development, but also in the maintenance lifelong. So when we have an excessive amount of PMP22, this leads to accumulation in the Schwann cell, stress in the endoplasmic reticulum, dysregulation of myelin, and continuous demyelination, and the known neuropathy and the phenotype of CMT1A. So most of the gene therapy developments for CMT1A really focus on targeting this gene dosage effect. So essentially, they try to reduce the levels of PMP22 protein. And this can be done at the level of the messenger RNA or at the level of the DNA. So at the level of the messenger RNA, we use microRNAs and short hairpin RNAs. These are small ribonucleotides that enter the cytoplasm as loop structures and then transcribe to mature structures to complementarily bind to the messenger RNA. We also have small interfering RNAs. These are more naturally occurring. They are already mature sequences, and they do the same. They specifically target mRNA sequences. By doing this, they conjugate with the RNA-induced silencing complex, and they accelerate the metabolism of mRNA and interfere with its translation, leading to reduced expression. Another approach is using antisense oligonucleotides. These are small nucleotides that recognize specific sequences on the messenger RNA. They go directly to the mRNA and block the translation of PMP22 protein. At the level of the DNA, so at the gene level, we have CRISPR-based approaches that target for editing, for example, enhancers in the PMP22 gene, such as the upstream data box, or other methods that bind promoter regions to reduce the gene expression. So these target the genome itself and the PMP22 gene. Beyond these disease-specific approaches, we also have some non-specific gene therapies that have been tried, for example, using hepatocyte growth factor or neurotrophin-3. These are supposed to improve myelination, but have a non-specific effect on Schwann cell biology. And we have also some downstream targets, such as the abolition of the purinergic ionotropic P2X7 receptor that is expected to reduce the pathology in Schwann cells. I will show you here the first example of the short hairpin RNA gene therapy that has been tried in CMT1A. This was the work by Nicolas Trigot group that was published a few years ago. So here the approach was by delivering an shRNA targeting specifically the mouse PMP22, and it was delivered into the CMT1A rat. This is a rat model that overexpresses the mouse PMP22, has several additional copies of the murine PMP22. The injection was done in a very early developmental stage at postnatal day 6. This is even before developing the neuropathy in this model. And essentially, they showed a very good expression and biodistribution in the injected nerve, which was quite localized, so there was no widespread distribution of the virus. They showed that in the nerve tissue, there is a significant reduction in the PMP22 expression. So here you see the CMT1A, which is untreated, and then the CMT1A treated with 2 different shRNAs is almost down to the level of the normal animals. So clear target engagement, reduction of PMP22 levels. At the same time, they showed that the myelin protein 0, which is the major myelin protein, was not targeted. In contrast, it showed improvement, reflecting improvement in myelination. And when they did the treatment trial, they also showed functional and morphological improvements. So there was improvement in the internode length, which is reduced in CMT1A models. So the density of nodes was decreased because the internodes became longer. The nerve conduction velocities improved compared to the untreated animal. Muscle strength improved. And then the morphology, as you can see it on the right side, you see a wild-type animal nerve. You see an untreated animal, many thinly myelinated and demyelinated fibers, and below the treated animals. And there was also improvement in biomarkers that were detected in blood. So overall, this is a proof of concept that if you silence PMP22 gene, you can achieve a real therapeutic benefit, but this is a localized treatment by intraneural injection. Another approach has been published by Budari et al. This is the Lilian Massad group. They use squalenoid siRNA nanoparticles. So this is a nanoparticle-based approach to deliver short interfering RNA. This was done by intravenous delivery, so it's a more systemic approach. And it was tested in mouse models of CMT1A in the JP18 and JY13 model. Again, as with the other model, they demonstrated that there is significant reduction in the level of PMP22 protein in treated animals. And there was also functional improvement, which was measured by grip strength. You see here on the right, improved force in treated animals as opposed to the dextrose treated. There was also improvement in the compound muscle action potentials measured in these animals and in the conduction velocities. See here after treatment. And there was also improvement in the myelin morphology in the peripheral nerves. This is wild type, this is the dextrose control, and these are the treated animals. So this is another nanoparticle-based approach to treat CMT1A more systematically. I will say a few more things about our approach. We also use a microRNA-based approach to target the PMP22 gene. So this is in collaboration with Scott Harper, who designed this microRNA to specifically target the human PMP22 transcript. It was first tested in cells for efficient silencing of both human and mouse PMP22, and then it was packaged into the adeno-associated viral vector 9 along with the control. And then we tested this therapeutic vector in the mouse model of CMT1A that overexpresses human PMP22. So that's the C61 heterozygous model. We showed with the reported gene that we achieve widespread expression of the vector after lumbar intrathecal delivery, both in the spinal roots and in the peripheral nerves. And we also demonstrated by real-time PCR efficient reduction of PMP22 mRNA levels in all peripheral nerve tissues. This effect was specific for PMP22 because other myelin-related genes, including MPZ, CNP, connexin 32, gliomedin, were all stable or upregulated. We also demonstrated reduction in PMP22 levels at the protein level in all peripheral nerve tissues. You see here lumbar roots, sciatic nerves, and femoral nerves. There is significant reduction in MER-treated animals as opposed to the control vector. And this effect, again, was specific for PMP22. The myelin protein 0 levels actually improved, reflecting improvement in myelination. So a specific silencing effect on the human PMP22. We then tested this treatment in the mouse model, both at early stages, but also at late stages of the neuropathy at 2 and 6 months of age, because it is important to test both pre and post-onset therapies. And we demonstrated with various behavioral test improvement in the motor performance in treated animals, both in rotarod and grip strength, and also in the mobility of the hind limbs. As you can see here, this is a treated animal, and this is an untreated or mock-treated animal. There was also improvement in the electrophysiology, both in the motor nerve conduction velocities, as well as in the CMAP amplitudes in treated mice, although the CMAP amplitudes did not reach the level of the wild-type animals. So this tells us that the correction is not complete. And we also had amelioration of neurofilament light levels and of growth differentiation factor 15, two biomarkers that were elevated in the model. We had similar efficacy of late treatment, both in terms of the nerve conduction velocity, but the CMAPs did not show significant improvement, and the blood biomarkers also did not respond as with the early treatment. Again, emphasizing that the earlier we treat, the more likely we're going to have a bigger therapeutic effect. The therapeutic effect was also shown with morphological analysis, and we found improvement in myelination. You see here a treated animal, and this is the mock-treated, so many more completely demyelinated or thinly myelinated fibers. These are the lumbar roots, and the same results in the femoral motor nerves. We also demonstrated in this model that there is a baseline inflammation, probably a secondary effect of demyelination, and this inflammation actually improves following treatment. So by measuring various inflammatory cells, B cells, T cells, and macrophages in the peripheral nerve tissue, we showed a reduction in the number in treated animals compared to the mock-treated controls. So overall, this was a proof of concept for an effective gene therapy to treat CMT1A. And this program now is moving towards translation. It's already being tested in non-human primates in collaboration with industry. So we hope it will proceed further towards the clinic. Here, I would also like to highlight another gene therapy approach, which is likely to be the first one actually moving into the clinic. This is the fatty acid ligand conjugated oligonucleotide-based approach, also known as Falcon technology. This was developed by DTX Pharma, and if you heard the news, it was acquired by Novartis this past summer. And basically, it relies on a technology where the microRNA targeting the PMP22 is conjugated to a fatty acid, and this facilitates the entry into the Schwann cells. So using this approach, the scientists of DTX Pharma showed that they can have therapeutic benefit in the CMT1A mouse model with remyelination of axons, improved muscle and motor performance. And they have also completed the GLP toxicology studies, and they completed also the investigational drug application filing with the FDA. So if everything goes well with this development, we may see first in human clinical trials for CMT1A by next year, which is an exciting development. So I will now like to switch over to the second most common type of CMT, which is the CMT1X. This is the second most common type. It is an X-linked disease, which results from mutations in the connexin 32 gene, the GJB1. It accounts for about 10% of all CMT patients worldwide and causes a mixed axonal and demyelinating phenotype with intermediate nerve conduction slowing. You see here in this diagram the normal expression of connexin 32, which forms gap junction channels traversing the myelin sheath in the area of the non-compact myelin at the paranodal loops and Schmidt-Lantermann incisors. In these areas, the channels serve important homeostatic roles, so they are able to transfer small molecules, including glucose, metabolic precursors, ion channels, and second messengers that are vital for the integrity not only of the myelin sheath, but also of the axon itself. So by studying various models of CMT1X, we and others have shown that in the absence of these gap junction channels, we have disturbed homeostasis in the peripheral nerves. We have beginning degeneration of the myelin sheath, but also simultaneously axonal degeneration with abnormal density of the neurofilaments, slowing of the axonal transport, and eventually demyelination as well. And you see here a typical electron microscopy image of a myelinated fiber with all this degenerating process in the innermost part of the myelin, vacuoles, and detachment of the myelin sheaths. So there are more than 400 different missense or other types of mutations reported to cause CMT1X. They're spread all over the protein, which is a typical transmembrane protein here. And so a lot of work has been done to understand the disease mechanisms by expressing connexin 32 mutations initially in vitro in cells. We and others showed that most of the mutations result in a retention of the protein in the cytoplasm and failure to form gap junction plaques, as the wild type protein does. The same happens also in vivo when you develop a transgenic mouse model. The knockout model of the disease, which is deficient for connexin 32, develops a very typical for CMT1X pathology with progressive demyelination and axonal pathology. So loss of connexin 32 is enough to cause the disease. And the knockout does not differ from the transgenics in this regard. We also know from a lot of clinical work that was done in series of families over the last 2 decades and published most recently by Record et al., that there is no typical phenotype-genotype correlation, regardless of the type of mutation, including a complete deletion or non-coding mutations. We have a similar phenotype, and the severity increases with age, indicating that overall we have a loss of function mechanism that accounts for CMT1X. So based on all this knowledge, the strategy here has been to replace the GJB1 gene in Schwann cells. Before describing our approach, I would like to also give you an overview of emerging treatments for CMT1X, because there are several efforts. The first, of course, is the gene therapy, and we work on disease-specific gene therapy to replace the connexin 32 gene, but there is also non-specific gene therapy using trophic factors. And there are also drug-based therapies that are under development, including targeting of the inflammatory cells. This is work from Rudolf Martini and now from Abram's lab, using either macrophage, colony-stimulating factor inhibitors, or inosine administration. There are also other approaches to prevent axonal degeneration or provide metabolic support, and these are all at the experimental stage. I show here very briefly the intramuscular neurotrophin-3 gene therapy treatment that is being developed by Professor Zachen's group in Ohio. So here, the approach is to deliver to the muscle a gene therapy that expresses neurotrophin-3. This is a neurotrophic factor that is predicted to have a beneficial effect on nerve pathology. And so when they injected the connexin 32 knockout mouse, they demonstrated improvement in motor performance and nerve conduction velocities. And there was also some improvement in nerve myelination and also in the degree of denervation in the muscle. So this may be an indirect approach to achieve at least a partial therapeutic effect. What we have developed is an AAV adeno-associated viral vector-mediated gene replacement for connexin 32. So here we package the coding sequence of the human connexin 32 gene under a myelin-specific promoter, the myelin protein 0 promoter, in order to achieve specific expression of the connexin 32 in myelinating Schwann cells. So we delivered this by intrathecal injection, similar to the 1A model. And when we delivered that into connexin 32 knockout mice, we demonstrated that we have expression of connexin 32 in the paranodal myelin areas of the lumbar roots and sciatic nerves, similar to the wild-type mouse as opposed to the negative control. And this was detected also by immunoblood analysis. And so using this approach, we tested first therapy in the connexin 32 knockout mouse. We used pre-onset treatment at the age of 2 months and also post-onset treatment at the age of 6 months. And both treatments resulted in functional improvement measured by the foot grip strength, so the muscle strength, but also with improved sciatic nerve conduction velocities. Again, as with the other models here, if you treat late, so after the onset of the neuropathy, the correction is less pronounced and the conduction velocities do not reach the level of the normal mouse as they do in the early treatment. There was also improvement in the morphology of the peripheral nerves. And because, as I told you, we have many different connexin 32 mutations, it is important to test this treatment in different models of CMT1X that express different types of mutations, because this is what patients will look like. They will have a mutation in their Schwann cells. So we tested the gene replacement strategy in 2 transgenic animals that express CMT1X mutants on a knockout background, the R75W and N175D transgenics. We showed here that despite the presence of the mutated protein, we get the virally expressed wild-type protein in the areas of non-compact myelin, and we performed similar to the knockout, the pre-onset and a post-onset treatment trial as well. And I will summarize very briefly just to tell you that we had similar therapeutic benefit, both in terms of the foot grip strength and the sciatic nerve conduction velocities in both transgenic animals before onset, but also when treating after the onset of the neuropathy. And we also had morphological improvements. These are examples of the spinal roots. You see here treated animals and untreated animals. There is improvement in the myelination. There is also a reduction in the number of macrophages, which are typical in the 1X models. You see here a huge foamy macrophage, and these macrophages are reduced after treatment in both mouse models. And the same effect was also observed in the distal femoral motor nerves. So this program is also moving towards translation, is currently now being tested in a dose escalation study and also in a non-human primate study to demonstrate the scale-up potential. So we hope it will find its way to translation as well in the near future. What we also try to do, and this is relevant for all CMT types, is to develop treatment-responsive biomarkers because they are needed for assessing the efficacy of treatments. One of the first biomarkers was the neurofilament light chain. It was shown by Sandelius et al. To be elevated in CMT patients with different types of CMT, including 1A and 1X. And so we evaluated these neurofilament light levels in our models, in the 1X model and in the 1A model. The neurofilament light levels are elevated compared to normal animals. And when we treat the animals, we see an amelioration in treated compared to the mock-treated animals. So this may be potentially a treatment-responsive biomarker. The only problem is that, at least in patients, it has not shown a reliable longitudinal progression, but it may be a good biomarker for a short-term treatment response. There is more effort to develop biomarkers for CMT neuropathies in blood, and this is work done by the group in Cambridge by Professor Horvath. And they also looked at various other blood biomarkers, including neural cell adhesion molecule 1 and growth differentiation factor 15. Both of them show elevation in CMT patient sera, and they correlate with the severity of the neuropathy. There is also another biomarker, the TMPRSS5, which may be more relevant for Schwann cells because it has been shown to be elevated in CMT1A. And so we evaluated both the NCAM1 and the GDF15 in our models, and we showed again elevation and amelioration after treatment, the NCAM1 in the 1X model and the GDF15 in the 1A model. So these are important developments that may be useful for the future clinical trials planning. I will move now to talk a little bit about some axonal CMTs briefly. Axonal CMTs in general are more challenging because they're mostly dominant, resulting from toxic gain of function, so they need more complex gene therapy approaches. One of the examples is CMT2D, which is about 0.4% of all CMT cases, so relatively common, results from dominant mutations in the glycyl tRNA synthetase, the GARS, which is a homodimer and charges tRNA with glycine. So in CMT2D and in the disease models, it was shown that the mutant GARS likely has aberrant interactions with other proteins, most notably the TrkB. And TrkB is a receptor that binds the BDNF, an important trophic factor, and which is present at the neuromuscular junction. So disturbed signaling through the BDNF has been speculated to disrupt protein synthesis and impact axonal transport. So the strategies for treating CMT2D are mainly based on trying to deliver allele-specific silencing. So this was published by Rob Burgess' group a few years ago. So this is an allele-specific microRNA gene silencing approach. They tested that in two models of CMT2D. Both of these models express mutations that consist of several base pair difference from the wild type allele. So the one is a 4 amino acid deletion, so 12 base pairs difference, and the other one is a five base pair difference from the wild type allele. So it was relatively straightforward to design an allele-specific microRNA targeting. And so they designed these microRNAs, they delivered them into the two models of CMT2D, and they demonstrated improvement in function and improvement in the motor nerve morphology in these mice. When they, however, and this worked only when they delivered it very early by intracerebroventricular injection, not when the treatment was delivered later on by intrathecal injection. And this relates also to the fact that with progressive axonal loss in axonal neuropathy, we expect a lower therapeutic benefit. Another important point here is that it is a challenge in general to achieve sufficient allele specificity when we only have a single base pair mutation, which is the most frequent scenario with dominant CMT types. So beyond the larger deletions causing CMT neuropathies, it will be more challenging, and we will need more sophisticated approaches to treat dominant disease with allele-specific silencing. But these approaches are also in development. The same approach that I described before for 1X has also been tested with neurotrophin-3 injection in the 4D, in the CMT2D model. So this is again the neurotrophic 3 vector injected intramuscularly. And when Ozes et al. Tested that in the model, they demonstrated improvement in neuromuscular junction innervation, in the electrophysiological parameters, and also in the morphology of the nerve and of the muscle. So that's, again, a non-specific approach to treat axonal neuropathies as well. Another example for CMT2 and basically relating to targeting motor neurons is the SMA with respiratory insufficiency, the SMART1, which is an allelic disorder to CMT2S, which is a later onset axonal neuropathy. SMART1, of course, is a very early onset severe SMA phenotype. So a gene replacement therapy for the responsible gene, the IgHMBP2, was developed by Shababi et al. This is a gene replacement therapy now because SMART1 results from a complete loss of function of the gene, and CMT2S results from a partial loss of function of the gene. So the approach is to replace the gene in neurons. And for this, they use the AAV9 vector with a ubiquitous promoter to deliver the gene. And they injected animals at very early stages, at postnatal day 1 to 4. They demonstrated that they can get a dose-dependent rescue of the survival. Here, the interesting part was that if you use a higher dose, you actually have a worse outcome on the survival. And the lower dose had a better effect on the survival, which creates also a note of caution when we replace genes, that we need to get the dose right, and that too much of a therapeutic gene may actually have detrimental effects. And of course, they showed at the morphological level improvement in the number of motor neurons in the spinal cord, and also in the neuromuscular junction innervation. So I will close with a few examples for CMT4 neuropathies. CMT4C is an early onset demyelinating neuropathy. This is autosomal recessive, loss of function, demyelinating CMT type, resulting from mutations in the SH3-TC2 gene, which is an important endocytic recycling compartment protein in Schwann cells. There is a model of the disease that we have used to develop a gene replacement therapy for CMT4C. We developed a therapeutic vector to deliver the SH3-TC2 coding sequence under a minimal version of the myelin protein 0 promoter, and we injected the mouse model of the disease, and we demonstrated expression of the SH3-TC2 gene in myelinated Schwann cells in the spinal roots and in the peripheral nerves. And using this therapeutic vector, we demonstrated both with early as well as delayed treatment in this model to have therapeutic benefit in the behavioral outcomes, in grip strength, and also in the nerve conduction velocities and in the CMAP amplitudes. This is work recently published. And we also saw improvement in the morphology of the peripheral nerves, so improvement in the thickness of the myelin sheath and lower numbers of demyelinated fibers, both in the lumbar roots, but also in the femoral motor nerves. Finally, we also evaluated the nodal pathology in CMT4C models. This is characteristic also of patient biopsied nerves. There is an elongation of the node of Ranvier, which likely contributes to the slow conduction velocities. The same occurs in the mouse model as opposed to the normal model. And so we looked in treatment groups whether we can remodel the nodes of Ranvier, and indeed we achieved to reduce the length of the node of Ranvier that was elongated in the untreated animal. This is the treated animal, and the quantification shows also improvement in this aspect. So the last quick mention is about CMT4J. CMT4J is one of the most severe CMT4 types. It can present also with an early SMA-like phenotype, and it results in mixed demyelinating and axonal pathology. We have loss of motor neurons in the spinal cord, but also demyelination in the peripheral nerves. So it's particularly challenging disease. And Presa et al. Developed a gene replacement therapy for the gene, the FIG4. This was delivered very early in the pale tremor model of the disease at postnatal day 1. And they demonstrated improvement in survival, motor neuron numbers, and myelination in the peripheral nerve. Here, the challenge was that the treatment had to be given very early. If they treated later on, so after postnatal day 7, survival was still prolonged, but incomplete, and the function was not fully restored. So again, indicating that the earlier the treatment, the more likely we're going to have a good outcome. So I will summarize the CMT gene therapy developments. I have shown you several examples of treating demyelinating CMTs and axonal CMTs with disease-specific, but also non-specific approaches and using various delivery methods. We also have various technologies, including gene silencing, which is something really important for CMT1A. Allele-specific silencing will be needed for some of the dominant forms, gene replacement for the loss of function disorders, gene editing is another approach, antisense oligonucleotides and non-viral methods are being developed. I will close with some of the challenges we all have to keep in mind when we move forward with CMT gene therapies. Many of these relate to the preclinical development. First of all, we have to use models that reproduce the disease phenotype. We have to test the treatments in more than one models. We need to use translatable delivery methods and vehicles that can be applied in patients. We need to be sure about the therapeutic target, that it will address the disease mechanism. We have to achieve adequate biodistribution. We have to think about the right intervention age. And we have to make sure that the development relies on no off-target effects and solid pharmacokinetics and pharmacodynamics. When we move to clinical translation, it is important to keep in mind that the earlier the intervention, the more likely we're going to have a beneficial effect. CMT neuropathies are disabling, but not life-threatening in most cases. So we really need to have very safe treatments. in order to have them accepted by regulatory authorities and by patients themselves. CMT neuropathies are very slowly progressive, so we need to have very sensitive, sophisticated clinical outcome measures and treatment-responsive biomarkers. And this is work that is being done in parallel with the gene therapy development. And we definitely need natural history studies for all CMT types, as they will be the basis for designing therapeutic trials and for successfully testing the treatments. I would like to thank you for joining the webinar and for your attention. And I would like to thank the members of my team for doing some of the work that I showed you from my lab, many collaborators, and the organizations that fund our research. Thank you very much.
Thank you very much for this very comprehensive review of many exciting developments that we start to see for diseases that a few years ago seemed to have no solution. So we have a question from Borja Cabal asking if you reduce the PMP22 expression for CMT1, if you risk to induce an HNPP phenotype.
Yeah, this is a very relevant question. Thank you. Of course, everybody thinks about the possibility that we will have excessive silencing leading to the opposite phenotype of HNPP. We have tested that actually in the model. When we treat the overexpressing mouse, we only see normalization of the levels. We never go too far down with the silencing. It looks like Schwann cells have a regulatory way to avoid excessive silencing. So we never see detrimental effect in the overexpressing mouse. We also tested the silencing in normal mice that do not overexpress the protein. We don't see much of a pathology there, but with time, you do see some functional defects, indicating that if you start from a normal level of the protein and bring it further down, maybe then you can have a detrimental effect. But it's certainly something that we need to see in patients. We don't know how patients will react over a long period of time, and we don't know how a patient with sick nerves over many years will react to the silencing. But overall, we don't get too much silencing and we don't get excessive silencing. And I think the main challenge is to get the silencing to the cells and not that we get too much silencing in Schwann cells. But it's a very valid argument, and we keep that, of course, in mind. And we look for that possibility in the testing.
I noticed that in your first mouse model for treatment of CMT1A, that the recovery of the nerve conduction velocities was very remarkable and very impressive. But on the contrary, the amplitude was not. And I was wondering if in a demyelinating disorder where the patient, the mouse, has early access to therapy, if there are any way axonal changes that you've identified as correlating with that finding.
Yeah, I mean, for sure, in all demyelinating neuropathies, including 1A, is the secondary axonal loss that leads to disability. And we know that develops over time. We all know that in CMT1A children, when you do a nerve conduction study, there is slowing. So the velocities don't really change over the lifespan. The demyelination is there from the beginning, from childhood. And as time goes by, the Cmaps go down, which means we have progressive axonal loss. So in the ideal world, we should treat children with CMT1A, because at that time, we can fix myelination, we can fix Schwann cells and prevent axonal loss. In reality, if we treat adults, we realistically would not expect that we can restore degenerated axons. So I think that's the reason also in the mouse that the later we treat, the less benefit we get on the Cmap. We still fix the myelination, but you can only preserve the remaining axons. You cannot bring back degenerated axons. So that's important to keep in mind also with the clinical translation point of view.
Excellent. I pass on to David for closing remarks and ending this webinar. Thank you very much.
Thank
you,
you.
Antonio. Thank you, Kleopas, for this very comprehensive and clear presentation of a complex topic. And just one quick question about intrathecal delivery. You nicely showed that there is widespread expression of the connexin 32, for instance, or other supplemented replaced genes product. What about upper limbs? Because you examined mostly lower limbs functionally and morphologically. Did you have some evidence of improvement in the upper limbs, for instance?
Yeah, that's a very relevant question. Actually, in the mouse, we haven't looked much because we don't have much functional readouts from the upper limbs, from the front limbs. So, but I can tell you we have first indication from the primate study that we get equally good biodistribution in the forelimbs. And so when you deliver intrathecally, you get, spread of the virus all the way to the cervical segments, and you can get equally good biodistribution to the ulnar and median nerves as you do with the sciatic and femoral nerves. So I believe by targeting the CSF intrathecal space, we can get the roots along the spine and both the upper and the lower limb nerves.
So this is very important.
But I think,
exciting.
this will be really answered by the primate studies because they're also, closer to the human body. The mouse is very difficult. It's a very small size and it's tough to know what goes through which route to the nerves. But the primate studies will be crucial.
Thank you very much. We exceeded a bit our time, but it was worth it.
Yes, it was.
Hopefully, we'll have you soon with us again. And next week, we'll have another CMT webinar, the last one of this month. So don't miss it. Please register, and we'll be back here to meet with you and discuss these exciting themes. Thank you.
Thank you very much.
Thank you.